Dr. Robert Malone, thank you for your time today. And I have a few questions for you. You are known to be the inventor of mRNA technology. Could you tell me and the viewers briefly what that means?
Thank you. So there was I was right at the center and seminal in a series of discoveries that happened in the late 1980s at the Salk Institute. And it was when I was a graduate student. I was only 28, 29 during that period. But it happened. It resulted in a large number of patents. Those patents were filed by both the Salk Institute and by a company that I left the Salk and joined shortly thereafter and brought all my technology reagents and protocols. And we had other discoveries, follow-on discoveries there. And it was at that company, Vical, that the use of RNA and DNA for vaccination purposes was first reduced to practice. It resulted in a large number of domestic US and worldwide patents. And those patents were used quite aggressively by Vical and then by Merck, which licensed that whole patent estate to prevent others from entering the field for the 20-year life of the patents. So often the question comes up, how come there was this big gap in commercial development between when you had your discoveries in the late 80s and the current situation? And the truth is that Vical and Merck, although they were not able to actually make a product themselves, and I was long gone at that time, I was an academic by that point, but they were never able to develop anything commercially after spending literally over $1 billion. But they kept everybody else from doing anything. And then when those patents expired, suddenly the field opened up.
I see.
So that was a little over 10 years ago. So that's the story. And it's not my fault that I was an industrial scientist at the time when those patents were filed. I got a total of $1 US for compensation. But the people that held the patents used them to prevent others from commercializing the tech. And that's the core story. There's a lot of details around that, but I think that's good enough for your viewers.
Yes, thank you for that. Recently, you've gained a lot of attention as you've raised concerns regarding the safety of mRNA vaccines. What are your exact concerns? And how are these vaccines different to conventional vaccines?
So what happened and I'm going to reel back to this origin story at the Salk Institute. There was this, at the Salk, both the RNA and the advector technology originated. So that's the J&J and the versus the Oxford AstraZeneca vaccines are both adenoviral vectored. Wow. And then currently we have the Pfizer and Moderna mRNA vaccines and CureVac coming up in the rear with their data, which unfortunately the dose was too low on those studies. But it's the same technology platform. The Salk lab where this happened was Inderverma's lab, a brilliant, and at the time, the leading researcher in his laboratory complex in gene therapy. And they originate from a study involving gene transfer of cells that had been genetically engineered back into mice and expression of a transgene protein, this is the words we use, in those mice. And the mice would stop producing the protein after about 3 weeks. And it turned out that the reason for that was because they were mounting an immune response against them. So this was the first of my big aha moments, was to make lemonade out of lemons. And instead of focusing on gene therapy as the application, classic correction of genetic defects in children, for instance, we could take that same technology and use it to elicit a vaccine response.
Right.
The senior postdoc in the lab left and created a company called Crucell.
Right.
And came to me a few years later and said, Robert, you're right. I need to pivot Crucell from a gene therapy company to a vaccine company. That was sold to Johnson & Johnson, and that gives rise to the J&J vaccine, same technology platform. So both adenoviral vector and mRNA vaccines trace back to a gene therapy lab, and they are explicitly gene therapy applied to vaccines. Important concept. People don't like to use those words because they think they evoke a visceral response in the population, in the public. But in the Security and Exchange Commission filings for both Pfizer and Moderna, there's explicit statements that acknowledge that these are gene therapy based and that the FDA perceives them as such. Okay, so core thing, they're both gene therapy technologies. In the case of the RNA, would you like me to give your viewers a short version of what this tech is and how it works?
Yeah, I mean, if you could be very brief on that, yeah.
So the metaphor that I use that helps people to understand it is, if we think about this whole system of the cell like it's your laptop computer.
Right.
Okay, so there's the metaphor as the laptop computer. Your genes, the genome, your chromosomes, are a hard drive in this metaphor. Okay, the programs on your computer are the way that information is coded into the DNA.
Yeah.
Okay. And I've used the metaphor of an industrial robot, like you can use to build cars or carve wood or anything else. Okay, so multi-purpose industrial robot is the equivalent of the protein translation machinery out in the cytoplasm of the cell. RNA is the bit stream that goes from your hard drive to control the industrial robot.
Right.
In this metaphor. Does that make sense?
Yes.
Okay, so DNA makes RNA, RNA makes protein.
Yeah.
The protein translation machinery is what is being kind of co-opted in your cells to actually manufacture the vaccines, both the adenoviral and the RNA vaccines. The RNA in this metaphor is a code that a hacker is inserting into that bit stream to make those robots make something they wouldn't otherwise make.
Yes.
Does that make sense?
Absolutely.
Okay. Awesome. So you've got it. Now you understand RNA vaccines and adenoviral vaccines. And in the case of the RNA, it only has to go into the cytoplasm. It only has to cross one cellular boundary to get in.
Right.
And it makes protein for a fairly short period of time, and then it gets broken up.
Okay.
In the case of the adenovirus vector, those are engineered to make lots of protein for a long period of time, and they're DNA based.
Right.
The adenovirus vector is a virus,
Yes.
cold virus, that's engineered so it goes into your nucleus.
Yes.
It doesn't insert into the chromosomes, but it stays in the nucleus, makes lots of RNA. That causes those little industrial robots called ribosomes to make lots of protein for a long period of time. So that's the big difference between the two tech, in a fundamental way, is the RNA makes the protein for a limited amount of time, and the adenoviral vector makes it for a much longer period of time. Now, what is it that they're making? We call this the payload.
Right.
And that is where things get interesting. So it's not necessarily that the tech, everybody focuses on this lipid or whether to the polyethylene glycol, or there's a whole bunch of details about the specific chemical composition of the RNA complexes that are used.
Right.
But what this distracts everyone from is the universal nature of the payload, the thing that's being expressed. And the thing that's being expressed in all of these systems is the spike protein.
Right.
Okay, so all of these genetic vaccines, which is what they are called, it's a category, they're genetic vaccines, express the spike protein in one form or another.
Right.
Which is essentially derived directly from the genetic sequence of the virus. So they're expressing a part of the virus, the knob part that sticks out from the surface of the virus.
Yeah.
And not the rest of the virus machinery and things that are necessary for the virus to replicate. Okay, that all makes sense?
Yes. Yes.
So that if you kind of get that into your head,
yeah,
then it becomes a little easier to make sense out of the data that are coming out of the adverse event databases.
Yes.
Okay, I'm getting techie again, forgive me. Okay, but we have to capture somehow this information about after people get vaccines, they report that they've had symptoms.
Right.
Something that they wouldn't normally expect. And those go into these adverse event databases. And then we have to use statistics to analyze those. And we have to do it really carefully because we have to sort out what's called confounding and masking and other effects that can make it so that it appears that there is some phenomena happening there. And it may not actually be true. It may be just an artifact of other kinds of noise. So
Right.
this analysis is actually quite challenging to do well. And it depends on how well the database is built. Okay,
Yes.
now let's park that for a minute
Yeah.
and answer your question, how are these vaccines different from other vaccines?
Mm.
In a regular classical vaccine, let's say influenza, you take a virus and you replicate it to high levels in either cell culture or chicken eggs, typically.
Yeah.
Okay, it'll grow in chicken embryos. And then you take that, you purify the virus away, and you inactivate the virus in some way. You may break it up into its pieces, but you end up with a preparation that's quite pure of, of, of pathogen particles of
Right.
some type, okay, that are killed.
Right.
You mix that with something that stimulates your immune system, and that's the jab you get.
Yeah.
Now, from a regulatory standpoint, you can precisely calculate and characterize exactly how much of the protein that goes into your shoulder.
Yeah.
In the case of these genetic vaccines, you really can't do that because you can precisely characterize how much of the genetic gene transfer material goes in your shoulder, but you can't really characterize very well how efficiently those go into your cells, the human, right?
Right.
And you have to try to extrapolate from animals, but that's not necessarily predictive.
Right.
You also can't calculate exactly how long they produce this protein, how much protein it makes, and
Mm.
exactly what cells in your body these things go into. So you end up, yes, you have little manufacturing factories in your body, but you may have a much more efficient gene transfer, and the kinetics, the timeline and levels that are produced in your body may be completely different from my body.
Right.
And you may have more sensitivity to something like coagulation problems
I
than
see.
I do, or I may be more sensitive to than you.
Mm.
And all of these things add together so that we end up, everybody understands the bell curve,
Yes.
right? A normal distribution of gene expression and of adverse events in a population.
Right.
So that's how we end up with only some people are experiencing some more severe symptoms, and most people are having a more average experience. And some people may have very little protein expression and very little adverse events and might even generate a suboptimal immune response. It's just the nature of the statistics of what happens with people. And this system of using genetic vaccines creates more variability in that than the classic method where you're just getting a jab with a very fixed amount of protein mixed with a very fixed amount of adjuvant that's very predictable. Okay, so
Right.
that's a fundamental difference. The other big one there is that the jab is not going into your cells, it's going out around the cells. Okay,
Mhm.
with the gene therapy technologies, the target is your cell,
It's going into the
cells in
cell.
your
Yeah.
body to co-opt those to become the little manufacturing factories that we talked about. Does that all make sense?
Yes. No, it does to me. Would you say, um, I mean, obviously, we've had UK was actually one of the first countries that gave their approval for the Pfizer vaccine. That was on December 2nd, 2020. Would you say that they perhaps maybe rushed through their approval of the Pfizer vaccine?
I wouldn't say maybe, I would say they did. There's lots of statements by various government public health authorities that they didn't cut any corners, but it's incontrovertible. You can't take a process that normally takes a decade or more and push it down into 9 months and not cut corners. And the simple math for the easiest one that everybody can see, is normally we wait at least 2 years after phase 3 when we have a large number of people that have accepted the test vaccine to make sure that there's no development of autoimmunity or neuropathy or other effects that happen with vaccines at a certain rate. All key principle is, I'm not an anti-vaxxer. I've spent 3 decades building vaccines and vaccine
Yes.
technology.
Yes.
And this is kind of my tech.
Yeah.
So I have a bias. If I have a bias, it's towards the tech, okay?
Exactly. Yeah.
But in normal situations, all vaccines have adverse events. All drugs have problems, right? You can
Yeah.
read the little package insert, and by the time you get down to the bottom, you won't think about taking that drug because you're going to be sure it's going to kill you after you read all that stuff. Vaccines have to be super, super safe because they're going into people that are otherwise healthy.
Right.
And so that's why we
Especially
have to
children.
have it. Well, there's that. Okay. And if you're going to open that door, that's where it really gets pretty dicey.
Yeah.
So here's the tie-in. All vaccines have some level of adverse events.
Right.
And what matters is how frequent and how severe those are, the bad things that rarely happen, compared to how frequent and how severe is the disease in an age cohort.
Right.
Okay. And so that involves a formal process of risk benefit ratio analysis,
Yes.
which is usually based on the actuarial table tools that the insurance industry uses.
Right. Yeah.
Quality adjusted life years. But in order to do the quality adjusted life year risk benefit calculations, you've got to have a bunch of data. You
Yeah.
can't just pull things out of the air. So you have to have data in order to make informed scientific decisions. It's evidence-based medicine. And what we've done for understandable reasons initially is we've said, okay, we have a global crisis. We have to do something. There was a bias worldwide that the something to do was vaccines as opposed to drugs.
Yeah.
So we pretty much put all of our eggs in that basket. That's a mistake. We could talk about that, but that's what was done for various reasons.
Yes.
So they put all their eggs in that basket and they said, we have to do this. We have to do this as quick as possible. And so we're not going to do all of the testing and all of the details that we would normally do.
Right.
Now, another thing happened during this rush. The regulatory agencies typically have checklists.
Yes.
They have a checklist for vaccines and they have a checklist for gene therapies. Those checklists are different.
I see.
With vaccines, for instance, you typically don't do what's called genotoxicity studies.
Right.
And you typically don't do reproductive toxicology studies.
Right. Which I was going to talk to you about that, about the documents released in Japan.
Just so I thought you might go there. I didn't know we were going to have time or not. So I'm setting that up a little bit. So typically those are done for gene therapy products,
right,
but not done for vaccine products because vaccine products are usually this kind of a standard jab that I was talking about.
Right.
Okay? And so those things didn't get done, or to the extent that they did get done, they got done under not in a very rigorous way. Okay.
Right.
And for the regulators, apparently, globally, everybody agreed that's okay. We're going to just treat these using our vaccine checklists, and that's all we're going to ask Pfizer to do. That, in my opinion, was one of the big mistakes. I think that a lot of this confusion that we have can really be traced back to the regulatory agencies and their willingness to suspend a lot of their normal practices and rigor in the face of the crisis.
Right.
And I think we can all understand that if we reel back in time.
Right.
to when our hospitals were full and our doctors were getting sick and people were dying and there was, talk about death on the street and all kinds of horrible things as if it was
Yes.
Ebola.
Right.
It
So,
turns out that it really wasn't that bad, in maybe in a lot of ways, and it especially wasn't that bad for children.
right. Right.
And so the thing is, that was then, and this is now. Back then, we didn't have the information that we have now about things like the severity of the disease in different populations,
Right.
is stratified by age and immunodeficiency and pregnancy and those kinds of things.
Right.
So we kind of applied, I like to say, if you give a 3-year-old a hammer, everything becomes a nail.
Yeah.
And that was, if you've had children, you understand what I'm saying. And so that was kind of what happened. But now we have a moment. Now you guys are in the midst of the Delta
Yes.
and that attack rate. Here in the States, it's currently only about 20% of cases are Delta. So
Right.
we haven't been hit as hard as you have.
Yeah.
But still, I think we now have enough data that we can go to a more evidence-based medicine approach.
Right.
Sense?
I just, sorry to interrupt you. You've mentioned a few times in previous interviews that we can't ignore the data. What do you think the data is telling us, and should we be worried?
So worried can be a strong word, and it can be misinterpreted, and it can be
Concern.
clipped out of anything I say and become a little tiny GIF that is thrown back at me and used
Okay.
to justify the BBC whacking me. So I'm going to use a
Should,
different word.
yeah, concerned maybe?
I think so. This goes my safe place to talk about this is to speak about bioethics. That's why I always talk about the bioethics. Okay, so we have these fundamental principles of bioethics for experimental products and experimentation on humans. Rule point number one: we're still in the experimental phase. These products are not yet fully licensed. They
Right.
don't have the data to support full licensure.
Right.
Since they're not yet market authorized, I think is the word you use in Europe.
Right.
Therefore, the bioethics are those which apply to human medical research. There's 3 core principles. Number 1, there has to be full and complete disclosure of risks. Point number 2, those risks have to be communicated in a way that you can understand them. We can't hide them using medical talk and sciencey talk. They have to be in plain language and stated in such a way that we can verify that you comprehend those risks.
Yeah.
Number 3, there has to be full, willing acceptance of the medical product voluntarily. No coercion,
Right.
no incentives,
Yes.
no free ice cream,
Yeah. Or fries or
okay?
burgers.
And I think pretty much all throughout the world now, we've already passed the coercion line
Right.
because we have all of this communication coming from public health authorities worldwide saying you have to take the jab. And often it's wrapped around the logic, if you don't take the jab, you're going to make Aunt Mary or Grandfather Jones more likely to die. So you have a public responsibility.
Yeah.
And if you deconstruct that, what we're saying is that we're okay sacrificing the right of the individual for the right of the community
Yes.
protected. That's the essence of it. So let's deconstruct that just a little bit more. The people that are at highest risk for disease and death are the elderly, full
Yeah.
stop. It's
Yeah.
the elderly, the obese, and some special populations.
Yeah.
Now, here's the inconvenient truth buried within that. The youth have a very, very low risk of hospitalization and death, particularly the adolescent, child, and infant populations.
Yeah.
Also, young adults have I'm talking about below 30, very low risk of hospitalization and death.
Yeah.
Okay, not that
I
they
think
don't get infected.
I think it's like 0.1% of
Or
COVID,
lower.
maybe
Yeah.
probably even lower.
Or lower, depending on where you are in that age range. Okay,
Right.
so it's
Yeah.
pretty tiny.
Yeah.
Okay, what we're doing by saying this, one hammer approach to everybody,
Yeah.
everybody needs to get the jab, is what we're doing is we're putting our youth at higher risk for vaccine related events. The ugly truth is that our youth will have some degree of disease and death attributable to the vaccine
Yeah.
in order to save the elderly.
Yeah.
Okay, if you really tear this apart,
Oh,
okay,
yeah.
that's what we're saying is we're okay as a culture
Yeah.
with putting our youth at risk in order to save our older population. And that, if you, that's why I don't want to ascribe intent, but if one does the quality adjusted life year calculations, that will become very, very visible because the youth have much longer lives than the elderly. And we do that calculation and do it for the risk benefit evaluation,
Right.
it really biases
Yes.
towards safety in the youth.
And also, we have no idea about any of the long-term effects
Well put.
you know.
Perfect segue. All we know is an emerging hazy picture.
Yes.
It's like your fuzzy, blurred out background of
Yes.
what our more acute adverse events are. We
Right.
have no idea about the long-term ones.
And what do you feel? I mean, only yesterday, Pfizer announced that they have plans to ask the FDA for emergency use authorization of its vaccine for children aged 5 to 11.
And
What's
so
that?
I'm very acutely aware of that, and I'm being contacted by various law firms regarding their interests in both the US and the UK regarding this issue. Um, uh, I can't ascribe intent by Pfizer in this. I can only say that the current data that we have available does not appear to support a favorable risk benefit analysis in that age cohort. Those are carefully selected words, but I think you understand the meaning.
Yes, yes. No, I do. Going back to these adverse events, what would you say are the sort of top three that you're finding looking at the data?
So what, and it's not me looking at the data, it's my colleagues at
Sure,
FDA and CDC
absolutely.
and independent analysts that are looking at the Israeli database that's more
Yes,
comprehensive. And now we have data coming out of Norway and out of the Netherlands. And I suspect that we will get the most rigorous, and I hear the yellow card system and
yes,
Tess Lori is raising some red
that's
flags
in
about
the
the yellow
UK. Yeah,
card.
we have a yellow card system.
Um, so in the Scandinavian countries, historically, because of their more fully integrated socialized medicine system, they have a history of being more rigorous in capturing these adverse event relationships.
Right.
And so when I was contacted by an investor group the other day asking for advice about these topics for obvious reasons, and I said, what you guys need to do is keep your eyes on the databases in Scandinavia,
Okay.
because that's where we're going to see the most rigorous data on adverse events relating to these vaccines, in my opinion.
Um,
And we're already seeing some of those data coming out of Norway. And
yeah,
as you know, or because you're right across the pond there from them, right across the channel,
yes,
you know that the Norwegians are starting to get a little worked up over some
yes,
of those adverse events,
yes,
and they are rejecting some of the vaccines, at least one of them, I
right.
believe, that is
Yes.
licensed in the UK. So I think that we have that I referred to the our ability to perceive events as a kind of like the blurred background behind you. What we have is incomplete, foggy data, and we're trying to derive meaning of that. So with all that, what seems to be the signals that are embedded within those data starting about 2-3 months ago, there was awareness that there was a signal of this set of cardiotoxicity events. So this is the
Right.
pericarditis and myocarditis,
Myocarditis, yeah.
and that has been seen most clearly in the young.
Could you just explain, just to, that is, I'm assuming that is the inflammation of the heart. Am I correct
Yes.
in interpreting?
Inflammation and fluid around the heart, and there's
Right.
various hypotheses about why that may occur. My favorite one is that we're getting what's called microcoagulation in the small vessels of the heart. That is one of the potential causes of that, but that's consistent with the biology of this virus. One of the core
Yes.
problems with COVID is coagulation.
Yes, the
Okay?
spike proteins. Yeah.
And you jumped right to it. Yes, it seems to be attributable to the spike proteins in the disease.
Yes.
And in the basic research, by the way, the spike proteins also open the blood-brain barrier, so that relates to some of the central nervous system
Yes.
findings that some people experience, including the brain fog.
Yes.
Okay, so those things would not be independent of age typically as findings. Okay, now one of the things about the toxicity in the heart is we're able to see that in the data in adolescents, in part because adolescents have such a low background event level of these kinds of heart problems. You almost never see them. So if you start seeing
Right.
them, it's
Okay.
easy to see the signal.
Because I was going
It's
to
really
ask.
harder. It's harder to see the signal in adults because we have heart problems in general. And so there's a lot more noise to that signal. To that end, there was a paper just out about these same kinds of heart problems in young adults and older adults in the US military.
Yes, I read that. I think Reuters had published that either
Well,
today.
they didn't publish it. They may have covered it.
They
It was
covered
in
it,
a
I mean,
peer-reviewed journal.
they published the article on it. Yeah.
Yeah, I think it was in the Journal of American Medical Association.
Yes.
But in any case, that's now out, and I predict that over time, because that those signals have been hidden by this confounding variable with masking problem that occurs with these databases. I talked about the database
Yeah.
analysis earlier. Okay. Now, within those databases also are signals having to do with thrombocytopenia. That's a big fancy word. What it means is low blood platelets.
Right.
Low blood platelets also relate to coagulation problems. So
Mm.
those are interrelated. And it could be spike related. The FDA is aware that thrombocytopenia is one of the known effects of administering oligonucleotides, fragments of DNA or RNA. And so it could be that thrombocytopenia could be partially driven by this unknown mechanism that's associated with administering polynucleotides, but it could be just a simple explanation that it's driven by this spike-mediated coagulopathy or coagulation problems. So
Right.
we've got now on our punch list, we've got the blood clotting problems,
Yes.
which are not trivial. We've got, and all of these may be interrelated. We've got the blood clotting problems, we've got the platelet loss, we've got the cardiotoxicity. There are others that are within there that aren't being brought out yet, and some of them relate to female reproductive health.
Yes, this was, I was going to ask you about this document released from Japan, the Pfizer report. Could you elaborate a bit on that, please?
So that's before we go there, can I just go at the top level?
Yeah.
I don't know about you if you've had your jab, and I don't want to seek medical information from you, but I bet in your world, if you talk to 15 or 20 different premenopausal women of various ages that have had the jab, many of them are going to tell you that it's disrupted their menses. Some of them are probably going to tell you that they had very heavy flow associated with that. Something's going on.
Right.
Now, there are data about the issue. Let's stay at the top level rather than going way down in the weeds of the Pfizer report quite yet. Okay, so what we're talking about is, are any of these concerns about reproductive health, are they clinically significant? Okay, it's easy for us scientists to get down into the weeds and have our little arguments about this, that, and the other thing. But it's all an argument about angels on the head of a pin unless it makes a clinically significant difference. That makes sense?
Yes.
So are there any information about reproductive health and pregnancy in associated with these vaccines? So there was the MMWR report from CDC based on the vSafe database, which is another one of these self-reported databases that are grossly underreporting events. And their conclusion was there's no signal there that they can see that suggests that there's a problem with taking vaccine during pregnancy. But they also say the data are so small and so weak that our assessment could change anytime.
Right.
And so that's not very encouraging.
Yes.
Then the NIH announced that they're doing a prospective trial.
Yes.
They just announced it last week.
Yes.
On safety of pregnancy. Well, some of us might say that would have been good to start that about 9 months ago.
Sure.
But now they're doing it. There are some data suggesting that there's no effect of vaccine on spontaneous abortions.
Yeah.
But those data are biased because they have a large number of participants that took vaccine in the 3rd trimester. And in the 3rd trimester, as you know, spontaneous abortion is quite rare.
Yeah.
It usually happens in the 1st and 2nd trimester. And if you take the 3rd trimester data out and just look at those patients, those women that were pregnant that took vaccine in the 1st and 2nd trimester, suddenly it's a different picture. And the spontaneous abortion rate, these are small numbers. This is an alternative data analysis. It's not the one that the authors published. They published looking at 1st, 2nd, and 3rd, with majority of patients being in the 3rd trimester. But if you pull those out and reanalyze the data, then your spontaneous abortion rate goes up to something north of 50%.
Right.
That's worrisome.
Yes.
Okay? So if you were to ask me today, based on the little tiny preliminary fragments of data that I have, is it safe to take vaccine during the 1st and 2nd trimester? I would say I would avoid that if I was you. If it was my daughter, I would say try not to get pregnant during this particular period, or get vaccinated before you get pregnant. But if you're already pregnant, I would probably wait at least until the 3rd trimester until you took vaccine. And I think that's a safe and responsible position.
Right, right. Going back to, I have a quick question. You, in your interview with Brett Weinstein,
Yes.
you'd mentioned that you had alerted the FDA for months before about your concerns with these vaccines. What was their response?
It was, so I have a small group of very senior people that are in senior offices within the FDA. We're basically old gray hairs. We talk to each other by Zoom every other week or so. And but they're in influential offices, but they're outside of the review branch.
Right.
Okay? One of them is within the review branch, but is so senior that she's now like an emeritus person. And so as this progressed early on last fall, and the trials were moving, I was acutely aware of the fact that spike has biologic activity outside of just being a receptor binding protein, because these viruses, these little RNA viruses, pack all kinds of activity in each and every protein. They don't just do one function. So for instance, spike manipulates the cellular immune response, essentially interferon production and other things, to the advantage of the virus. It's one of many viral proteins in SARS-CoV-2 that do this, okay? So spike is not inert. It's not an inert antigen. It has biologic activity. And so in speaking to them, I said, hey guys, I'm not sure that anybody is taking into account that spike is not just an antigen. That's how the developers are approaching it, but it has other biologic activities. And they said, prove that, show me the data. Okay, good position, solid position to say. And so I sent them papers that have been published both on SARS-CoV-2 and on SARS, because they're very similar, that showed that spike in those and in coronaviruses in general have a variety of biologic activities, including this kind of immune manipulation. So I sent those in,
Yes.
and they said, okay, we're going to forward that to the review team. And then the next time we met, they said, well, they looked at that, but they weren't convinced.
Right.
And so they really don't think that there's enough information there to raise any alarms or concerns.
Right.
Now, in retrospect, that was not the right decision. And this gets to, I like to say, so I've been fact-checked by Reuters, but by the way, Reuters a board chair also sits on the Pfizer board.
Yeah.
So I've been fact-checked by Reuters always, and they've asserted that these statements about spike being cytotoxic, having direct toxicity, are false.
Right.
Now that was easy for me to refute. They relied on the sources of government officials, and they kind of twisted the words of those government officials. They didn't directly address my point. Spike is cytotoxic. The native spike is cytotoxic.
Right.
That is in multiple peer-reviewed publications, not my publications. I'm just reporting what's out there. So many people have verified this using a variety of systems. So it's not fake news.
Yes.
For whatever. So the native spike is, here's my point about this, and you'll understand it. Pharmaceutical development is like the French judicial system in terms of safety. You're guilty until proven innocent. The way that things work is it's Pfizer's job, not my job, to prove that their version of spike is not toxic.
Right.
Okay?
Yeah.
And I haven't seen those data.
Yeah.
I don't know that they exist. And I think in this rush, what I think I see, I speculate, is there's always this fog of war and kind of a wartime mentality. And sometimes people are willing to overlook things that they might be more meticulous about when we didn't have all this pressure.
Right.
And my reading of that Japanese document in broad sense, and I don't read Japanese, but if I also combine that with the authorization letter, the justification letter from the EMA, I just had an unstable connection. If I combine the Japanese document, which has data tables in English, as you know, and other data that is in English in the figure legends, but the body of the document is in Japanese,
Yes.
and I can't read Japanese.
Yeah, I saw that.
But if you combine that with the EMA statement, and I have the Pfizer protocol, clinical protocol. Pfizer says explicitly in their clinical protocol that the reproductive toxicology has not been done. So it's not my words, I'm not speculating, it's documented.
Right.
Likewise, in the EMA document, it's quite clear that this reprotox and genotox have not been performed. In the Japanese document, in the English data tables, there are some data that are quite, have raised concerns, even alarm,
Right.
among some of us, including me. And what they show is that these key studies that are required at a preclinical stage, one is required to characterize using the active drug. So the thing that you're going to inject,
Right.
must be characterized rigorously for where it goes and what it does. Okay, it's fundamental, makes sense to me in animal models, so rodents, at least. And it has to be done in a rigorous sense, and our fancy words for that is good laboratory practices. So these are standard international norms for how these types of studies have to be done.
Right.
The studies that were done were not done according to good laboratory standards. They were non-GLP. Warning flag number one.
Right.
Warning flag number two, the studies concerning how much protein and where. So this relates to the fact that it's a gene therapy product and it's causing cells to make the protein, the antigen.
Yeah.
They didn't actually look at spike antigen. They looked at RNA that was coding for a surrogate, the protein that makes the firefly tail glow. It has the unfortunate name of luciferase, and in the States, this evokes certain visceral responses from some religious communities um, because of the reference to the devil. But it's just the name they gave to the firefly protein. It has nothing to
Right.
do with the devil. So this firefly protein can be used really sensitively as what we call a reporter gene, because you can put the RNA coding for this. And this is what I did in my initial discoveries, by the way. They actually were pioneering in use of luciferase for looking for gene expression of RNA. It'd never been done before. And they still use the same method. And the way that it's usually done to get very rigorous assessment of how much protein is being made is that you dissect the animal, you take tissue samples from each of the key organs, you put them in a tube, and you do some tests, and you get a very precise number of how much of photons that are produced from
Right.
this light emitting reaction that can be converted into precisely how much protein is there that was made by the cells that got the RNA.
Mm.
Now, there's kind of a little bit of a parlor trick that can be done with this, because it's such a great system that you can take a whole animal after you've delivered the gene and inject the substrate, luciferin, and put a photon camera up above it. And you can see those photons, that glow, coming out of the animal.
Right.
Okay. And it has to go through the tissue and the bone and the muscle and the skin and the fat and everything else and the fur. And then it gets to the photon camera. Okay. What happens is that those photons get scattered all over the place.
Right.
And so if you use the whole animal imaging, you're only detecting the areas that have the most luciferase.
Yes.
Pfizer, for some reason, used this method, which is the least sensitive method, to look at where the protein's being expressed. So not only did they not look for spike expression, which is what they could have done, they used a surrogate, luciferase, and then they used the least sensitive way to detect the luciferase. And they said, oh, look, the protein is only expressed at the site of injection.
I see.
Okay.
Right.
That's kind of cooking the books.
Yeah.
Just a little, like a lot.
Yeah.
And bad on the regulators for not catching that.
Right.
Okay. Now they also did two other experiments. So there's two other things. You're looking at, that's for looking at where the protein is expressed, but they're not actually looking at spike. They use a surrogate and the least sensitive way to detect it. Then they also did a very sensitive assay for where the RNA goes. They put a radioactive tag on it. And in that case, they did do the right experiment and dissected out the tissues and analyzed them for the RNA signal.
Right.
And it clearly shows that after injection, most of the RNA stays at the injection site, but a lot of it goes all over the body.
Mm.
Yes, some of it goes to the ovaries, but not so much as the lipid part, which is a separate assay they do in the same way. So they, if just reeling back for your viewers, if we imagine this system is like a letter with an envelope.
Yeah.
The letter inside is the RNA. The envelope is a cluster of fats that coat the RNA. Okay, and it's the letter envelope is what gets the RNA into the cell.
Yeah. Just
Okay?
going back to the messenger RNA, is the messenger part that, like the envelope?
No, the messenger, going back to the metaphor, remember the computer metaphor?
Oh, right. Yes, yes.
RNA, so the central dogma is DNA makes RNA, RNA makes
Yes.
protein. The RNA is like the bit stream that goes from your hard drive to the robot.
Yeah.
And it's a message.
Yes, that's message. Yeah.
Okay? It's a message. So there's other types of RNA. RNA is used for other things in cells, but one of the things that's used for is to convey a message.
Yes.
So that subset of RNA is called messenger RNA.
I see.
Good?
Yeah.
So going back to where we were, Pfizer also in that Japanese data package, which is historic right now, it was submitted presumably last fall. Hopefully they have a whole lot more data now. Hopefully they fixed the problems that existed back then. I can't tell. They didn't send it to me. They haven't shared that. They haven't published it. But in those historic data, with this laboratory assay that wasn't really very rigorous, so we got to remember that.
Mm.
Okay. What they showed was that the lipid part, the envelope that the RNA slips into, also distributes all over the body.
Right.
And what has some alarm, it concentrates in the liver. We'd expect that. That's liver's function is filtering blood. It goes in the spleen, and we'd also expect that because that's part of what the spleen does in a different way. But 12% of those lipids accumulate in the ovary, and they don't accumulate in the testes in the same way. There's also a higher amount of lipids and RNA signal in the bone marrow. Now, does that prove that spike protein is being produced in the ovary? The answer is no, 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 don't say that. A lot of people saying that on the internet. It doesn't, that is not what this shows. What it shows is that this untested lipid, not previously used in large numbers of humans, that's synthesized, does concentrate in the ovary. Now, is that clinically significant? I have no idea.
Right.
Because the reproductive toxicology studies weren't done.
Hasn't been done. Right.
Okay.
Yeah.
So is it holy moly, the house is on fire? No, we can't say that.
Yeah.
We can't say that there's a signal here of some concern that merits rigorous follow-up.
Right.
Does this mean that it has anything to do with the dysmenorrhea or other female reproductive symptoms or potential signals relating to spontaneous abortions in the first and second trimester? We have no idea.
So essentially, it's the concentration of these lipids in the ovaries that is atypical.
Is at the heart of this controversy about those data. And
right.
Here's what I'm trying to express is the caveat. We can't beat them up for not doing good laboratory practice assays that are rigorous, and then turn around and beat them up for data coming from non-GLP studies. We're trying to have it both ways if we go there. So I think what we have to do responsibly is to say that what we have are early data that were not rigorously captured that suggests that there may be a signal of lipid concentration in the ovaries, but that needs to be verified by true good laboratory practice studies.
Yes, it needs to be investigated.
And why, the 800-pound gorilla in the room is, why didn't all the regulatory agencies see this when everybody else sees it as soon as they see the package? And why didn't they light up and say, Pfizer, let's wait a little bit, send me the GLP studies, and then we'll talk, which is normally what they would do.
Yeah.
If I submitted that package for a drug that I'd developed that was outside of this, okay, and they would have said, no, go back and do your homework.
Right.
We'll talk to you then. So that's one of the oddities. And the other key oddity here is that the regulators, certainly in the US, had the right to insist that the pharmaceutical companies do much more rigorous capture of information during this emergency use authorization phase. And they neglected to exercise that right. That's the gentlest way I can put it.
Right.
And if they had insisted on exercising their right to require more rigorous data capture during EUA phase on safety and toxicity, then we wouldn't be having all these arguments, because we would have the data and we could analyze the data and we would know the answer, and we wouldn't have all this ambiguity about what are the adverse events. We would know by this point.
Yes.
And so both of these problems at the root, in my experience, if you let big pharma have their way, they will have their way with you.
Yes. And there hasn't been full disclosure, essentially.
Just so. And part of that, in defense of organizations like the CDC or the MRMC, perhaps, I speculate, uh, or whatever the acronym is for your medical research
We have,
council.
yeah, we have the MHRA.
MHRA. Okay, so in
Which I'm
defense of
actually
it,
I want to touch on the MHRA. You use the word vigorous a lot, in the sense of testing. And so the MHRA, they made their announcement that they'd authorized the use of these vaccines based on, well, following a rigorous review of their safety, quality, and efficacy. Do you believe that statement is untrue, given what you've just said?
I I don't know. I know the data package that was submitted to Japan, and I know some insider information about the FDA and what they did. I know for a fact that the FDA did not apply the checklist for gene therapies. They only applied the vaccine checklist.
Right.
Okay. And they may, so I can't speak of your regulatory agency because I have no intelligence, no data to speak to it.
Right.
But speaking, if we assume that this Japanese data packet represents a common technical document,
yeah,
and therefore the same data tables were submitted to UK, EU, and US and Canada, if we take that as the assumption, if they approved based on those data tables, I'm not convinced that there was a rigorous assessment. Right. Because it took me and another regulatory professional that was even more senior that I referred this to because I was upset I was afraid. When I read these and I was asked to draw conclusions, I drew my conclusions and they were frightening to me that I would be attributed to saying these things. And so I checked with another more senior person and he reviewed them, and he's the one actually that found the discrepancies in terms of the genotoxicity and the reproductive tox. I hadn't even realized that.
Right.
And he gave me the details of specifically which reprotox studies weren't done.
Yes.
Okay? So good on him. But if the two of us can pick this out in 45 minutes of review, I can't see how we could claim that there was a rigorous review. To say rigorous, rigorous is a term without any specificity.
Yes,
it's in the eye of the beholder.
How do you define that?
Yeah, it's in the eye of the beholder.
Yeah.
So how can they may have believed that they rigorously applied the standard vaccine checklist.
Right. Right.
I just can't. This gets to, I can't get in their heads. I don't know exactly what happened. I don't know what the transactions were, and I don't know what they were thinking. I only know what I can observe.
Yes. Just going back to, given that close to 4 million, I think it's 3.96 million people have died worldwide from COVID-19. The counter argument says that, the benefits of these vaccines, they outweigh the risks. And it's, the vaccines are our only chance of going back to normal. What do you make of that?
So let's dissect that, unpick that just for a moment. What you're talking about there is the aggregate mortality across all ages. And if you just take just the slightest little bit more granularity in those data, what you're really saying is we've had X, whatever the number is, millions of deaths.
3.96 million.
3.96. I'm willing to bet that at least three of those is going to be in people age 65 and above, probably even more. Okay, so that's a little bit of a sleight of hand to say it that way, because you're applying the death rate to the whole population, but the deaths are only occurring in the elderly for the most part, or people with some severe or significant pre-existing conditions. Okay. Yeah. So to apply that event across the whole population and then say, on the basis of what's happening to the old, we need to do this to everybody, that actually is not aligned with normal vaccination practice. Okay? Right. Normally, when we roll out a new vaccine, we do analysis for each of the key cohorts: elderly, infants, children, adolescents, healthy normal adults, immunosuppressed, and pregnant women. Each
Yeah.
of those are analyzed separately. Okay.
Yeah.
And we're saying, no, we're going to throw all that past practice out and we're just going to apply it to an aggregate. That's the first point.
Right.
Okay. Second point, and this obviously feeds back into justification for authorization for children.
Yeah.
The second point, the logic that we have to vaccinate the whole population in order to prevent this and get back to normal. Okay. Let's unpick that for a moment. What they're talking about is herd immunity. We've all heard of herd immunity.
Yep.
Okay. Herd immunity is actually a precise epidemiologic term. It's well understood. It is what happens. In order to reach herd immunity, you get to the point where if you're vaccinated, if you're infected, the probability that you will infect somebody else is less than 1. In other words, with a reproductive coefficient of 3, for every 1 person that's infected, they infect 3 other people. If we get to the point for every 1 person infected, they infect less than 1 other person, then the virus can't circulate anymore and it becomes extinguished, and we call that herd immunity. Herd immunity can be achieved by a large fraction of the population having already been infected. It can be achieved by vaccination if the vaccines are sterilizing or are blocking to a significant extent. Here's another technical term: transmissibility.
Yeah.
That has to do with that rate that I was talking about. What is the probability if you're infected? How many other people are you going to infect?
Yeah.
Okay.
That's the R rate, isn't it?
That's exactly right. And R naught is the baseline R rate with no intervention. And then R changes as we change our behavior, use social distancing, put on masks, get vaccinated, all kinds of things go into changing R. Okay. So good on you. In order to calculate the percent uptake of vaccine required to achieve herd immunity, we have to know the data on the impact of the vaccine on transmissibility. Does that make sense?
Yeah.
Because herd immunity is about transmissibility. We have to have those data.
Yeah.
Okay. We don't have those data.
You don't have the data. Yeah.
The data were collected. So when we hear 90% efficacy,
yes,
or whatever, right?
Yes. Yes.
What that is it protects to a very significant degree. It protects you from getting hospitalized or dying when you might otherwise have been hospitalized or died.
Right.
Okay? So those are disease endpoints. This disease is largely driven by your personal immune response against the virus.
Yeah.
It's not the virus that kills you. It's your response to the virus. It's this hyperinflammatory syndrome. Not everybody gets it.
Yeah.
Okay. So when you measure disease and death, it's only an indirect relationship to effects on the virus and the virus spread. Now, to this point, you in the UK know the people that have been previously vaccinated are getting infected with Delta.
Yeah.
And the World Health Organization put out a recommendation that we all need to wear masks again because of Delta. Remember?
Yeah.
Just a couple days ago. Okay?
Yeah.
Now, you all understand what this means. It means that your prior vaccination is not protecting you from being infected by Delta. And it's not protecting you from having virus replication if you get exposed to Delta. And it's not protecting you from shedding virus if you've already been exposed to Delta, which means you can infect other people. Okay, so how frequently? We don't have the data. I don't have the data. I can't tell you. But what I can tell you is if you ever hear a public health official say, we have to have 70% vaccine uptake in the whole population to protect us by achieving herd immunity, what you can immediately conclude is they don't have a fig of an understanding of what they're talking about.
Right.
Okay, because the data don't exist. We can't make that calculation. It's simple. Okay, so the point is, the vaccines are not what we call sterilizing. I'm not talking about your personal reproduction. I'm talking about the ability of the virus to reproduce.
Yeah.
So they are not sterilizing for the virus. You can still get infection. You can still get virus replication. You can still spread virus. How frequently? I don't know.
You don't know.
They do seem to be really good at protecting against disease and death. Yay, fantastic. Okay, so then they should be applied. Here's the logic. Ready? They should be applied to the people that are at high risk of disease and death.
Yeah.
Now, the logic that we as a culture, as a people, have to all take the jab to prevent disease and death.
Yes.
Let's pick that apart. What we're saying is we all have to take the jab with the associated risk because we're currently healthy in order to protect the old people.
Yes. Well,
Back
yes.
to that point, okay?
Yeah.
So the easiest way to protect the old people is to get the old people vaccinated.
Exactly.
Okay?
That's
It's
common
easy.
sense,
It's
isn't
a
it?
simple stuff.
Logic. Yeah.
Yeah, we don't have to apply. So remember what I said, you give a 3-year-old a hammer and everything's a nail. We
Yeah.
don't have to use that hammer on everybody,
Yeah.
the hammer being the vaccine. We only need to apply it to the people that are at high risk. And if Auntie May or Grandmother Smith doesn't want to take the vaccine for whatever reason she does, that's her personal choice. And she's accepting the risk. And it's not your job to take a product to protect her. It's her job.
Yeah.
That's how it works in a free society where we have free will, in my opinion.
Yes. Why do you think there is such a push? And obviously, we have the MHRA approval, the FDA approval for the vaccine for children between the ages of 12 and 15. We know the FDA, Pfizer has plans to ask FDA approval for children at a lower age, aged 5 to 11. Why do you think there's such a push to get the children vaccinated?
I don't know. And you're asking me to get into the head of Pfizer. A cynic might mention the financial compensation at stake. I wouldn't do that, but some might say that.
Right.
And so, this gets to how avaricious is a pharmaceutical company. Is
Yes.
basically what you're saying. That's for you guys to judge. That's kind of a political question, not a scientific question.
Yeah.
And it has to do with incentives, financial incentives, and other factors that are out of my control. But in terms of the underlying logic, which is what, again, this is my big message. Number one, that it's your choice and it's your body.
Exactly.
And you should not be coerced, because that goes back to the Nuremberg Code.
Yes.
Right? We all agreed on that. We had that discussion and we agreed, just
Yeah.
to reel back, not to say that public health officials are Nazis. Okay, don't go there. But if we go back to what happened with the Nuremberg situation, the fundamentals there was the argument that the Nazi regime made that the benefits to the many outweighed the benefits to the risks to the few. That it was justified to take conscripted individuals without their permission and subject them to cold treatment and other scientific investigations because the data were necessary to protect their troops in Moscow and Finland.
Right.
Okay, that was the essence of that argument, was the benefits to the many outweigh the risks to the few. Right. And we as a culture all agreed, no, we disagree with that. And furthermore, we disagree with it so strongly that we're going to hang all of you guys, is what happened, to be blunt.
Yeah.
Okay? So pretty clear that we all agreed on that. And so this argument that we all have to, now this, this is a very different situation in that the risks of the vaccine are not as severe. But nonetheless, we've all agreed they have to be fully disclosed. And if we're going to mandate or in other ways encourage or all the coerce, we can use stronger and stronger words.
There's bribery going on as well. I know in Greece they're paying young people to take the vaccine.
Well put. Okay. So that is not legal under US federal law. Now, whether or not they disregard that, there it is. Canada too. Canada rolled out the policy of ice cream cones to entice kids to get the jab without their parents' approval. That, I think we can all agree, is crossing a line too.
Yeah.
So, what motivates Pfizer? I don't know. It's the job of our public officials to protect the population and the individual. And
Yes.
I argue that they need to, buckle down, do their job, make the hard call based on evidence-based medicine, which we now have enough data to support. We know that there are risks in these populations. We don't know all of the risks yet, but we know enough of them.
I'm going to raise a question of conflict of interest here. I think it's pretty known that, I know with the MHRA, I'm not sure with the FDA, is that a lot of those senior officials are either ex big pharma, executives.
Or they're looking for their exit strategy to become big pharma or consultants to big pharma, right?
Right.
That's in DC, that is the standard way. You go to work for the FDA for half a dozen years, and then you get out and you join a consulting firm and you rake the bucks. That's
Right.
just the way things are in DC.
Yeah.
And might be the case in the UK, I don't know.
Yeah.
So the phrase for what you're touching on is called regulatory capture. And it happens in the States. It is a major problem in the States in
Right.
organizations like the USDA. The USDA is basically been run again and again by people that have strong ties to Monsanto. In the UK, you understand what I'm saying.
Yes.
So we call this regulatory capture, is the
Right.
term. And a case can be made that we've moved beyond regulatory capture. That's the point we seem to be, and this gets back to our first topic, the censorship.
Yes, which I really want to talk to you about. And I know time is going.
We seem to be in a situation where there has been integration at the top between large media
Yeah.
and pharmaceutical industry and government, and all the way up to WHO.
Yeah.
And the thing that, for me, it was perplexing. I couldn't understand. I could see the footprints. I could see the breadcrumbs and the signs, but I didn't know how this was happening. Until I was alerted about the Trusted News Initiative, which is led by the BBC.
Yeah, I've heard about that. Yeah.
And then suddenly everything kind of came together.
Yeah. It's sort of this combat against disinformation, misinformation.
Was originally designed for combating political disinformation around elections.
Right.
And under BBC leadership, I mean, this isn't a conspiracy theory. You can look up the BBC press announcement from last fall. Okay, they're very proud of this. Under BBC leadership, the Trusted News Initiative Cooperative, which cuts across all of large tech.
Yeah.
Microsoft, Google, Facebook, Yahoo, all that. Okay, so this Trusted News Initiative cuts across all of that. They're all linked together into a consortium now.
Yeah.
And they've all agreed that the only truths that will be allowed to be discussed on their platforms are those truths that can be tied back to key governmental agencies such as the CDC or the WHO. So the version of truth, of scientific truth, the only version that's allowed to be discussed even is those truths that have been endorsed by these large bureaucratic public health agencies.
Right. Given what you've just said, do you think, given that you've recently been locked out of your LinkedIn account?
Right. Not just locked out, I've been erased.
You've been erased, right.
And there's also erasure of my footprints even on Wikipedia. Yeah.
Wow. Wow, it's shocking, yeah. And also your interview with Brett Weinstein and Steve Kirsch was recently.
Now that's overinterpreted. So
right.
YouTube, we were able to track through the press because when YouTube drops you, in this case, Brett was deplatformed. So that means he's demonetized. I'm sorry. He
Right.
can't get revenue anymore. So then that's his personal family revenue is from his podcasts. He can no
Right.
longer get revenue anymore. So that's
Right.
what YouTube did. YouTube
Right.
has made statements to other third parties in the press that the reason why they did that was because Brett was talking about ivermectin, which he's a big fan of.
Right.
Okay. It was not because of me talking about Spike.
Right.
Okay. So good on YouTube, and don't overinterpret those data. It wasn't because of anything I said. And if you watch that video, which I find fascinating, 3 old men talking to each other for 3 hours and it goes viral. What is that saying? That's crazy. So it happened, but we had one of those gentlemen, Steve, who is really wound up about vaccine safety, and he overstates the data. He's very enthusiastic about it, but he has absolutely no background in biology,
Yeah.
let alone medicine, let alone drug development. He is an engineer.
Yeah.
His CV is that he has multi-$100 million from his discoveries involving engineering, like the optical mouse in Silicon Valley.
Yeah.
So he's enthusiastic. Brett is totally focused on ivermectin,
Yes.
and bless his heart for that. But it is what got him whacked. In my case, I received, as you've seen, this statement from LinkedIn about the justification for my being ghosted. And
Yeah.
just, it wasn't that they just closed my account. I no longer exist. You
Right.
cannot find R.W. Malone, MD on LinkedIn anymore.
Wow.
And I've had a presence there for at least 15 years.
Yeah. Wow.
So all of that documentation, photos from inside the WHO during the Ebola outbreak, all kinds of stuff, poof, gone.
Gone.
And their justification, as you saw when I forwarded to you that
Yeah.
document, has 3 main elements. One is that, so the things they cited as the justification for deleting me was some scientific discourse about the spike toxin in citing scientific literature.
Yeah.
A posting of the Wall Street Journal editorial, a section of that with
Yeah.
the title and a link that spoke to the risks of these vaccines.
Yes, I read that. Yeah.
Pretty mainstream press.
Yeah.
Not too controversial. And me talking about censorship.
Right.
And the problem with censorship. So I got censored in part for talking about censorship.
By talking about censorship. Yeah.
But the top, if you look carefully at the top, the first thing they cite, and the temporal relationship here, this is like deaths and vaccines. Okay, there's a temporal relationship. We can't prove a causal one. The first thing they cite is the LinkedIn post where I outed the chairman of the board of Reuters for having his relationship with Pfizer. Okay,
Right.
that is the top post that they cite as the reason for ghosting me. Now, is that cause and relationship? They didn't say, well, the really reason we did it is because you said something that you're not allowed to say. But clearly, that's the case across all of those posts that they cite.
Yes.
When other people have looked at that rationale, they're struck that there's nothing there that provides a rationale.
Yes.
So there's some subtext there that's being applied. It's not like I was flaming at somebody or attacking somebody or doing the other overt things. It was because I was saying speech that is not allowed.
That's not allowed.
And it was scientific speech, and it was speech about censorship. And it was specifically mentioning a chairman of the board of Reuters. Okay.
Right.
So, which is part of the Trusted News Initiative.
Yeah.
So, cause and effect, I don't know. But when people look at that, it's pretty bizarre, especially the fact that the subject line has misspellings in it. So this wasn't a template that was auto-generated.
Right.
Somebody took the time, apparently hastily, to write this. And I happen to know that Steve had called a buddy of his in Silicon Valley, who happens to be a vice president of LinkedIn. And that guy had asked internally, what the heck went on with Malone?
Right.
Okay, so I got deleted about within 48 hours of when I outed this board director from Reuters.
Right.
Then this vice president started asking questions. And
Yeah.
about 48 hours later, I get this memo with typos that somebody must have composed hastily, I infer,
Yes.
that has these specifics that I relate to you. So what it speaks to, as I think it's self-evident to all of us,
Yes.
is this integration between big tech, government, and biopharma that many of us are becoming quite concerned about. And I'm getting a lot of communication back from Europe and the States. But as I said, it's all the conservative press in the States. In Europe, it's intellectuals that have deep awareness of this vexing problem. What happened in Germany in the 30s? How did these intelligent, well-educated people go so sideways? And I think the best explanation that a lot of people are coming up with is it's fear. Fear drives groups of people to do things in moments of stress that they wouldn't otherwise do, to cross lines and agree to do stuff that they wouldn't do in a normal situation. And I think that what we're seeing is a distortion of political behavior driven by fear. And the more insidious question behind it is, are there people that are exploiting this window
Yeah.
for their own purposes?
Yeah.
Whether it's financial, political, power, and that, as somebody involved in public health, that would be a huge travesty. Most of us are working our cans off for little compensation. The repurposed drug work that my team has done, we've intentionally not patented anything. We want it in the public domain. We want it to be able to be used in Sri Lanka or any place else without any royalties.
Yeah.
And if there are people out there that are exploiting this for financial power or political reasons, that's just not, talk about not being in the common good.
Yes.
That, I think the expression, it ain't fair, it ain't right, it ain't proper.
Yes. Oh, really, Dr. Malone, thank you so much for your time today and for speaking to me so candidly and explaining things so clearly.
Thank you.
I really appreciate your time.
I've tried, and thank you for such a lovely morning spent. Let's. I'm going to go have my coffee now.
Good. Enjoy your coffee. Take care.
Thank you.
Thank you so much. Bye-bye.
Likewise. Bye-bye.